Okay, so this is the last lecture of quote-unquote new material before the midterm. Next Monday we'll have a review, but there will be a couple of clicker questions based on this lecture that we're doing today. One quick question, and I have an extra recorder up here. Would it be useful to people to have also MP3s, just audios of lectures? Yes, I'll try and get that and post those as well. I'll just post them on the D2L link for that. It's relatively straightforward for me to do that. Again, all relatively straightforward. I'm trying to post all of the videos together again on YouTube. That, for some reason, last time was causing me all kinds of problems. Eventually, I figured out a way to deal with it. Hopefully, the next couple of lectures should be a lot more straightforward to do that. Again, the software is what's going on there. Of course, midterm, Wednesday, there's a quick question on D2L about this. It will be here, normal class time, starting at 9. You can have as long as you need. You need one of the big Scantrons, and I've been told those are the blue ones. I will make sure that the number for that ends up on D2L, and I'll also mention it again on Monday. 50 questions, multiple choice. Other questions about the exam at this point? Yeah? Can we get the Scantrons at the bookstore? Where can you get the Scantrons? I believe you can get them at the bookstore. I don't need them, so I don't get them there. But hopefully some of your colleagues will know. So you apparently can get them in Smith, just downstairs in the little kiosk. Right by the cafeteria, apparently, at that little store. Yeah, we have the stationery. You can get them there as well, and the big ones. Yeah, full-size page ones. Other questions, comments, worries? Yes?
Are your clicker questions similar to how you write exam questions?
So the question is, are my clicker questions similar to exam questions? Yes. And that's partly why I do clicker questions, is to give you some of that idea. Also, almost all of my old exams are on D2L. There's an insane number of them. It's a little hard to find them. Sorry about that. But I just put them all up there, the exams and all the keys. Yeah? Will I be posting the clicker question I've done throughout? If I have time after I'm done writing the exam, yes. But I make no guarantee that I'll be able to do that. Yes? They are all on the recordings, yeah. So if you just want to go and look at the recordings on YouTube, they're the first 5 minutes of each class. They're there. So again, it's just a case of me going through and pulling them all out. Yeah?
You said that we used this book in a different year. What year was that to look at the exams?
Oh, right. So there's a question about how to compare exams, and that's a great question. Thank you for bringing that up. Because last year when I taught this course, I used a different textbook, and so the syllabus got moved around a little bit. So if you look at last year's exams, they're not going to correspond completely to this past year's exams. So the fall 2012, because I usually teach this course in the fall, that's going to be the one which is most similar. That's when I last used the previous version, in fact, of the textbook for this. And the syllabus is really quite similar to that. There are a couple of minor changes. And so if you want to take a look at that, the syllabi are also posted in each of those. Everything is on D2L. Other questions, comments, worries? So speaking of clicker questions, let's go ahead and get started with those. If DNA replication were exclusively dispersive instead of semi-conservative, after 3 generations, Mielsen and Stahl would have seen blank bands in their experiment, 1, 2, 3, 4, or 5. Again, feel free to discuss this. That's a big difference from clicker questions and the exam questions. You're not allowed to talk to your neighbors during the exam. Seconds. 10. Okay, this is what people think relative to this question. There are a couple of things that I did want to mention, and actually thank you for that question about whether clicker questions are similar to exam questions. If you're to go and look at exam questions, you're highly likely to see something almost identical to this. Oops, let's go back. As far as questions are concerned, I didn't go back and look at those old exam questions. I didn't pull the clicker question from there, but these are things that I think about. So there are a couple of keys to this question. One is what the heck is dispersive replication? And that's okay. It's the individual pieces, individual pieces of the DNA, which are being copied. And so you've got a mixture of old and new in your replicated DNA. Then the next part of the question is, what would you see with measles and install, which is separation based on density. And what this means, if you have dispersive replication, first round actually looks exactly like semi-conservative replications. You've got half and half. It's that second round of replication where you're going to have 1/4, 3/4, but each of them is going to be the same. All the 4 that you've replicated. For the next one, when you've got 8, it's now going to be an 1/8 and 7/8. So it's all going to be one band, and so you're never going to see a big difference. So 1 is in fact correct here. Second question, the enzyme that generates the template for replication is DNA polymerase I, RPA, topoisomerase I, topoisomerase II, helicase. $20. $10.
Will they all have a part in it? Helicase nicks and loosens, topoisomerase opens, DNA polymerase attaches.
Okay, so most of you think it's DNA polymerase I. I don't agree with you. So there are 2 possibilities. One, most likely it's a bad question. The second possibility is that I did a really crummy job of explaining it. So we'll try and go through that then in a little bit more detail. It's partly why I do these clicker questions. So the key to this question is, what's the template? What's the template for replication? If you think about what DNA polymerases use, what do you have to have for replication? You have to have a primer and a template. And what is that template? Okay, the template is not RNA. I think I heard it here as well. You need a single-stranded DNA. So how do you generate a single-stranded DNA? Okay, you break it apart. But what do you mean by breaking it apart? You pull the 2 strands apart. How do you pull 2 strands apart? With what? A helicase. So to generate a single strand, you need a helicase. And so the key here is, what's the template? Now, one could argue it's a not well-worded question, because how do you get that template in the first place? And what we haven't talked about, we're going to talk about a little bit more today, but I mentioned really briefly, DNA polymerase I has this really bizarre activity, which is a 5' to 3' exonuclease activity. So it chews up the DNA as it's making it. And because of that, it's not the replicative polymerase. If you had a replicative polymerase, that chewed up stuff in front of it as it was moving along, this would be incredibly inefficient. In fact, nothing would really happen. So the polymerase which does replication in E. coli, which is where DNA polymerase I is from, is DNA polymerase III. So that would have been a possible answer to the question, so it means I need to be careful about proofreading my exams, which I try. Yeah? RPA, replication protein A. What is RPA? SSB, single-stranded binding protein. And that is something that I would expect you to know. Okay, so the answer, the correct answer here is E. It's helicase.
So is that question null and void then?
No, that question is not null and void. But only 7 people got it right, and so hopefully when we normalize at the end, that will disappear. So that's the idea. And that's why I normalize, in fact. It's to kind of cover my posterior, because those bad questions, that means you actually can choose which are the bad questions. Those are the bad questions, the ones you missed. And the bad questions that you actually got right, you get to get count, as opposed to me throwing them out. Because if I threw it out and you got it right, the people who actually got that question right would be really annoyed. Correct? Yes, all 7 of you? Don't nod too widely, as people would know what it is. So, I'm ignoring that comment, by the way. DNA replication. This is exactly the same outline that I had before, with a little bit of highlighting on it, because we've talked about mutations, semi-conservative replication instead of dispersive replication, the mechanism, how do you do that, particularly the fidelity that you have of polymerization, which is checking your base pairs, and then the proofreading activity, which is what kind of exonuclease activity? Got 2 choices, 50%, pretty good deal. Is it 5' to 3' or 3' to 5'? So it's 3' to 5' because it's backing up. Your polymerase is always going from 5' to 3' because you're extending on the 3' ends. You've started at 5, you're going to 3. So the direction that you're going, 5' to 3'. Then backing up, proofreading is going to be 3' to 5'. So it's going in the opposite direction. So that's your proofreading. Replication fork, we're going to get into much more detail here. Again, the idea is you pull those 2 strands apart. What does that pulling apart? Helicase, exactly. Once you've pulled those 2 strands apart, you're now going to have 2 strands that have to be replicated. One's easy to replicate because, again, polymerase is going from 5' to 3', which means you're moving in the same direction as the fork is being opened. That's your so-called leading strand. But since you only have polymerases that go from 5' to 3', on the lagging strand, you've got to go in the opposite direction. And so that we'll look at in a little bit more detail. Somebody asked me about this right before lecture today. That's what's also known as semi-discontinuous replication. Some of you may have seen that in some of the slides or old exams. So the idea is discontinuous is individual pieces that you then end up putting back together. And it's semi-discontinuous because one strand is continuous and the other one is discontinuous. People call it semi-discontinuous. You could also call it semi-continuous. But just to be confusing, molecular biologists call it semi-discontinuous replication. To keep everything together, at least as far as the polymerase is concerned, you need the sliding clamp. We talked last time about how you get the sliding clamp onto DNA. You have to have a specific clamp loader complex. So that brings us again to what we're going to be talking about today, which is really the replisome, which is putting all of that stuff together. Molecular biologists love to use the word zome, replisome, ribosome, spliceosome, you name it, ome. But that's basically saying that you've got a whole complex of almost always proteins and nucleic acids together. So that zome, and it's actually something which is pretty big. You can see it in the microscope, electron microscope at the very least. So we'll look at that, again, what the components are of that replisome, and basically that's what you need to replicate the DNA. Then we'll talk a little bit about mismatch repair. This is how you get that final amount of fidelity that you have in your normal replication, getting to 1 mistake in 10 to the 9 or 1 mistake in 10 to the 10, depending on which literature you believe. Talk a little bit more about supercoiling. because it's so much fun to talk about. No, it's because it's really important in terms of replication. And so replication is, in fact, one of the places where you really do have to deal with supercoiling in a big way. A little bit about eukaryotes, and then where do you start, which I think someone brought up last time. So let's look at the replication fork. This is, again, where you're undergoing replication. This is your old DNA here, and these are your two new DNA strands at the back here. There's, again, 5' to 3'. So as soon as you have a polymerase which is going 5' to 3', this is really easy, and that's this leading strand here, your template, the old DNA, which actually it's easier to see here in yellow. I think HDMI is better than VGA, which is good. The yellow strand, that's your template strand. It's got a 3' end right here, the 5' end of the new one that you're making. That again gets extended by this replicative DNA polymerase. And that again in E. coli is DNA polymerase III, not DNA polymerase I, because it doesn't have this funky exonuclease activity, which is also going from 5' to 3'. All DNA polymerases love to fall off of DNA, so you've also got this sliding clamp that goes together with it. Yeah?
So you said it replicates 5 to 3. Are you referring to the template it goes 5 to 3, so it's actually making the 3-strand attachment? Is that what you're saying?
Okay, yeah, so 5' to 3' and 3' to 5' is always a confusing aspect here. And I always find it's really useful to write it out. And your exam paper, whatever it is, start with your 2 strands of DNA, write 5' at one end on one strand, 3' on the other strand. And so the template strand here, this end of the template strand is going to be the 3' end. The end now of the new strand that you're making, that's 5'. And so you're always going to have the 5' end pairing to the 3' end. So again, this polymerase is going 5' to 3' on the leading strand. If we had a polymerase that went the other way around, from 3' to 5', which we don't, then this would be fine. Now we have to deal with this lagging strand, and this is where the interesting part comes into the replication complex. It's made, again, in a semi-discontinuous fashion, and that means that this polymerase, which, just like that polymerase over there, is replicating from 5' to 3', is going basically the opposite direction relative to the replication fork. It has to, because it has to go backwards. Turns out, however, that everything is in one big complex right here, and this is that replisome. So you've got your leading strand polymerase and your lagging strand polymerase all together in one big blob, and we'll see that blob in just a second. Also, because you're making smaller pieces on this lagging strand down here at the bottom, you have to have a DNA primase that's making RNA primers all the time. Because each time you're making one of the Okazaki fragments, the discontinuous part, you have to be putting down an RNA primer. And of course, that RNA primer gets chewed up by DNA polymerase I and exonucleases in bacteria. We'll talk about what happens in eukaryotes in just a second. Yeah? And nucleic acids. So what's the nucleic acids in here? So the replosome is part of, any zomes are proteins and nucleic acids. The nucleic acids here, of course, are DNA. So that's what we have here. So as well as the DNA primase, you have a lot of single-stranded binding protein because the DNA helicase has pulled those 2 strands apart. This poor DNA polymerase over here is replicating as well as it can, but this helicase is going at 1,000 nucleotides a second. It's pulling this stuff apart. So you end up with a lot of single-stranded DNA. If you don't have single-stranded binding protein around, or RPA in the case of eukaryotes, then this has a big problem. So you've got a lot of that. Then, each time that this lagging strand polymerase gets to the end of one of these Okazaki fragments, basically right here, it's got to fall off the DNA and get back on, but get back on over here. How does it do that? It does that with the sliding clamp. How do you get the sliding clamp on? You have the clamp loader. So you have a combination. I'll get to you in just a sec. The sliding clamp is always associated with the replosome, and the clamp loader is also always associated with the replosome as well. Yeah, in the back. So where's the machinery fixing and joining the Okazaki fragments together? So the answer is taken off of this slide for simplicity purposes. And we'll see that when we look at an exploded version here. But the important aspect, particularly of this one, is to show that the leading and lagging strand polymerases are actually directly associated with each other. They're all together in one piece. And what that means is that the DNA is actually moving in and out of this complex. And people call this the trombone model. And we'll take a look at the animation for this in just a second, because I think it makes it a lot easier to follow. What it means is you've got this single-stranded piece, which is going to be single-stranded for a while until it gets copied, and then these things get chewed up at the end. Then it goes back and does the next piece. Yeah?
What did you say was moving in the opposite direction?
So if you look at where this polymerase is, it's also going from 5' to 3'. But if you think about this end over here, we had this end over here. That was the 3' end, right? This one started out paired to that. And so this now is the 5' end. So you can't replicate going this way because the polymerase doesn't do that. So it's got to flip around and go in the opposite direction. So that's what I mean by the opposite direction. It's still going 5' to 3'. It's just you have to flip the template around. And that's what this loop is all about here. Yeah?
In prokaryotes, for the latent strand, would all of that replication be happening by polymerase once, that it can just sort of keep going and chop through the RNA polymerase?
Okay, so the question is, in prokaryotes, would this guy down here basically be DNA polymerase I? The answer is in bacteria, because archaea are prokaryotes too, and they do it differently. So in bacteria, it's also DNA polymerase III that does this. DNA polymerase I is getting back to the question that we had in the back here. That's the one that comes in right at the end and cleans stuff up. It's not the one which is doing the replicative polymerization. Okay, so let's actually look at what this really looks like. Well, really looks like when you've separated everything else out. Here's a replisome. So it's got your DNA strands. This is what's being replicated. It seems that it's come off of the lagging strand a little bit here. And there's that loop that we'll take a look at again in more detail in just a second. We know a lot more about the structure of the E. coli reposome down here. This is a figure from the Watson textbook that, again, is on reserve in the library. You've got two DNA polymerase IIIs here, again, because the leading and lagging strand are being made by DNA polymerase III. So-called tau protein that holds everything together, and then this clamp loader and sliding clamp, which is there. Just another way to look at this, again, same process. Here we have our leading strand polymerase together with a sliding clamp. Here's our helicase that's separating these two DNAs, our primase here, and then our polymerase, which is extending in this direction. To risk beating the horse too much here, this is what you see in a eukaryotic system. Basically, exactly the same thing is going on in terms of this overview. But this shows a little bit more of that trombone model where you have the lagging strand polymerase that starts out here, again, connected to your complex, making an Okazaki fragment. Once it gets to the end, it has to fall off of the sliding clamp and go back and get onto the next sliding clamp for the next Okazaki fragment. And so what you end up with are a whole bunch of these sliding clamps on the lagging strand. And everybody remember what that sliding clamp is called in eukaryotic systems? Okay, well, what's the name? What's the name that people use for it? PCNA, proliferating cell nuclear antigen. Means that when you have replicating cells, you have a whole bunch of it. And that's mostly because it's bound to the lagging strand. Because you've got a lot of lagging strands around and a lot of this PCNA, which is actually associated with it. So getting back to your question, before I move. So the replosome usually is including DNA as well. Yeah, so again, it's the nucleic acid plus proteins.
So that third clamp is sort of there. Is that like on deck? It's going to be the next one.
Right, so you mean the third clamp? So the question is, is this third clamp here the one which is on deck? It's, it's ready to be jumping in when you get that. So what happens is, as soon as you have the primer which is formed, which is supposed to be this little red line on this slide here, then you get the clamp loader putting that clamp on. Of course, as soon as the clamp loader gets that, it's got to find a new clamp to put on when you're done with that next Okazaki fragment. And again, as we'll see in the animation, this happens really quite frequently. Okay, so getting back to, again, sorry, the question in the back that you had a couple minutes ago. What about all the other stuff which is in the replosome? Again, it's not just the polymerases and the clamp loader. We talked about the primase already. Here we have RPA, because it's just the names for the eukaryotic replication fork. PCNA, again, it's that sliding clamp. RFC is the replication factor C, horrible name, but it's the clamp loader. It's what puts that clamp onto the DNA. That's pretty straightforward for the leading strand. Also, there's a helicase. The helicase is listed here as being T antigen. That's actually not what you have in most cells. The only time you have this as a helicase is when you have a virus-infected cell. And that gets back to why viruses are so totally cool. And that's because you can use viruses to understand how cellular systems work. And until very recently, the only replicative helicase we knew about was actually a viral protein. Now we know that it's called an MCM protein. But for a long time, it was that T antigen here. So that's all in the leading strand, helicase, single-stranded binding protein, your polymerase, sliding clamp, and the clamp loader. This, of course, is attached to the lagging strand polymerase, which is here, together with sliding clamp and clamp loader, single-stranded binding protein. So that's exactly the same leading and lagging strands. Otherwise, I'll get this question in just a second. I want to finish this up real quick. So there are a couple of differences that you have now on the lagging strand. Of course, you've got to get rid of those primers. That's one aspect of things. But it turns out eukaryotes have another polymerase called DNA polymerase alpha, which extends primers. It's not clear why they have this polymerase that extends primers. Just that they do. It does seem to be important. You can't get rid of that particular gene. So it's sort of working like DNA polymerase I in bacterial systems, but it doesn't have that 5' to 3' exonuclease activity to get rid of RNA. And so in eukaryotic systems, you need some different proteins to do that. RNase H, which is in fact identical. You have RNase H in bacteria and in eukaryotes. And then this last protein right here, FEN1. So FEN1 stands for flap endonuclease. So we talked about exonucleases before. They chew in at the ends. An endonuclease cuts in the middle of a DNA. Well, what does it work on? It works on a flap. What is that flap? Well, the flap is what is generated when your lagging strand polymerase hits a RNA primer. What it does is it displaces that RNA from the DNA, giving you an RNA flap, as they call it. That gets chewed off by the flap endonuclease. And just as you have in bacteria, you need a DNA ligase to hook these ends back together. Now, there were two more questions here that I didn't want to skip over.
So I'm getting confused. So this is DNA polymerase III for eukaryotes and bacteria.
Okay, so eukaryotes. Just to make things ridiculously complicated for you, they use Greek letters for their polymerases. Whereas in bacteria, they use Roman numerals. So in the eukaryotic system, this is the Greek letters. So we've got Paul delta. Paul delta is the equivalent in bacteria of the eukaryotic DNA polymerase III. Okay, so the question is, do you have, and I'm paraphrasing here, do you have a single sliding clamp on the leading strand, or you have multiple sliding clamps on the leading strand? At least at the overview here, it looks as if you just have one. And that's going to be the one that you start with right when you start replicating and goes all the way through. If you go back and read the article that I got that figure from, the citation at the bottom, it says, well, actually, that's not completely true. You end up with a few, but way fewer than you have on the lagging strand. That's probably because you end up making nicks in that strand, and it's really important for repair of your DNA. And also mismatch repair that we're going to get to in just a second. So you have a few, but nowhere near as many of those pCNAs, at least in the eukaryotic system, on the leading strand as you do on the lagging strand.
If there isn't triphosphate at the end of the 5', how does DNA ligase do its connecting?
So the question is, if there's not a triphosphate at the end, how does DNA ligase do its job? You know, hook the two ends together, the 5' end and the 3' end? Well, if you think about it, the process here, which is making your RNA, if you've got an endonuclease, it's going to be chopping that off. So you don't have your 5' triphosphate, which is there. And usually, when you're making your extension each time, you're going to have then a monophosphate and an OH. And it's that monophosphate and the OH which actually get hooked together by the DNA ligase. It's not a triphosphate that gets added there. Yeah? Yes. So how are the leading and lagging strand polymerases connected? So I talked about the tau protein, which is what you see in E. coli. There are other proteins that I'm not going to overburden you with their names for, that are holding those two polymerases together in eukaryotic replication forks as well. This has basically gotten rid of those just so there's enough space for all of the names to fit on these individual proteins which are here. And speaking of names, this is a nice back of the envelope way of comparing these two. We've had a lot of questions about this already. What corresponds to what? So DNA polymerase III in bacteria, which is the replicative polymerase, corresponds to DNA polymerase delta in eukaryotes. So just straight across here. Bacteria, DNA primase, eukaryotes, DNA primase, and DNA polymerase alpha. So it's a primase alpha complex, which makes your primers. Your helicase in bacteria also goes by the name of DNAB. The MCM proteins are almost certainly, and again, there's a little bit of controversy about this, but it's pretty much been laid to rest now. It is the MCM proteins that are involved in this. SSB, as we talked about already, that's the bacterial situation. RPA, which is in fact a trimer in eukaryotes as opposed to a monomer that you have in bacteria. The sliding clamp is beta in bacteria. It's pCNA in eukaryotes, and the clamp loader is RFC in eukaryotes. So now I wanted to switch gears slightly here and take a look at this video. This is, again, it's linked. You can go to it through the PowerPoints. But this, I think, gives a really nice overview of, if it's actually going to start. No, we're not actually starting here. How nice. Okay, let's back up a little bit. There we go. DNA replication. The beginning of this actually goes over histone binding and DNA compaction, which is also quite useful if you want to take a look at it. So here's that replosome complex. It is simplified. There's no SSB. There are a number of different things here. But basically what we're trying to see here is leading versus lagging strand. So down here at the bottom, that is your leading strand polymerase with this sliding clamp on it. It just goes and goes and goes and goes and goes. So that's relatively straightforward. Here we have, in our light blue, this is the helicase that's pulling those two strands apart. Now we look at our lagging strand. So here's that lagging strand polymerase. It's polymerizing along until it gets to that next Okazaki fragment, falls off, and then this polymerase, which is still, again, attached to the replosome complex, will move over, bind to that next sliding clamp, which is appearing right here, and then get to the end of the Okazaki fragment and continue. And what that means is each time you have a loop now of this lagging strand, which of course is now coded in single-stranded DNA binding protein, which we don't see on this slide, and extends, gets longer and longer and longer until you start it again. So do you want to go then do that again? Yeah. Yes. Yeah. Question, why rewind? Those little clamps stay on there? Yeah, what happens with the sliding clamp is the question. They stay on. Eventually they decay. Eventually they'll come off. But it takes quite a long time. If you think about it, something which is literally a ring around your DNA is going to take a while to come off. So this is the, there's also a soundtrack that goes with this, so you can take a look at that there. So again, we've got our helicase, which is right here. Leading strand we don't have to worry about. But the lagging strand. So we've got clamp loader, which is a sort of gray thing that loads the clamp, which is this green thing, onto the lagging strand. That loads it on the lagging strand, and then the lagging strand polymerase will associate with that. Again, it just bounces back and forth here. There's your lagging strand polymerase, gets to the end, moves over, starts the next Okazaki fragment. Gets to the end here of the Okazaki fragment, moves over, starts again. Yeah?
So is the green the clamp and the dark purple the polymerase?
So it's green the clamp and dark purple the polymerase. Exactly, yes.
Is there no SSD on that animation?
Yes, the question is there is no SSP. There is no SSP in this animation. The only way to actually be able to see these things is to separate out these. Again, it's way more complicated than this. Plus, you've also got histones and everything else which is in there. But at least in terms of thinking about leading strand versus lagging strand, that's really what the point of this is about. Yeah?
The sliding strands stay on the lagging strand. Are those part of a marker for lagging?
So the question here is if those sliding clamps that stay on the lagging strand, are they a marker for the ligase? I wouldn't say they're a marker for the ligase or, again, fixing those problems. But they do associate then with protein-protein interactions with ligase and the Fen1 endonuclease, et cetera. And so there's some protein-protein interactions that are really important. Now, some people talk about PCNA or these sliding clamps as being the molecular tool belt of replication because they've got to interact with the polymerase, they've got to interact with the primase, they've got to interact with clamp loader. So they've all got these various different interaction domains that are associated with them. Yeah? Yeah. Yep. Good. Thank you. Okay. So let's move forward with this. So that whole replication process is wonderful. It's great. It's got these cool animations. Well, of course, they're completely oversimplified. And one of those major oversimplifications has to do with why I tortured you a week or so ago with supercoiling. And that has to do with your twist versus writhe. So twist is when you've got the two strands of DNA which are wrapped around each other. When you pull them apart, you're untwisting them. Well, if you're untwisting them, you've got to do something with that, all that extra supercoiling. So what do? You end up with writhe. What pulls the two strands apart? What pulls the two strands apart? Clicker question. Number 2, helicase. So it's the helicase which is doing that. It does this at 1,000 nucleotides a second. So you're getting a whole bunch of untwisting that's happening. Well, that whole untwisting is then leading to, in front of your replication fork, a whole bunch of writhing, where you're getting stuff which is really overwound. You unwound it here, it's getting overwound at the other end. So you have to get rid of that. If you don't get rid of this, and people have done some really nice experiments looking at this, the polymerase stops. You can't go any further. It just gets completely gummed up. So you have to have a topoisomerase to be able to deal with this. And again, in some of those replication forks, I didn't talk about it last time, but if you've been reading carefully, you notice that there's, in the replication fork, there's a topoisomerase, which is part of the replosome. It's right there. It's sitting right next to the helicase, which is getting rid of that extra superhelical tension as it's moving along. So this is the figure here from the Watson textbook. Another way of looking at this is from our textbook. Again, this is the experiment that was actually done. If you just take a helicase together with double-stranded DNA, put it on there, that helicase will untwist, 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 untwist until it just stops, because you can't get rid of all of these extra superhelical tension which is in front of it. How does that get taken care of? Topoisomerase. Two different flavors of topoisomerase. We talked about these before. Topoisomerase I, Topoisomerase II. Again, molecular biologists have no imagination. The easy way to remember this, however, were someone to ask you about it on an exam, is that Topoisomerase I cuts one strand, Topoisomerase II cuts two strands. So it's really quite straightforward to understand this. How does Topoisomerase I go about cutting one strand? It has a tyrosine side chain. So Topoisomerase I is a protein. It's got a very specific tyrosine. Tyrosine is wonderful because it's one of the 3 amino acids that has an OH at the end. And basically, what it does, it said, oh, this OH is going to act like a 3' OH in DNA. So it'll hook up to a 5' phosphate. And then once you have that interaction, it basically cuts the DNA, forms this covalent linkage. Now that it's formed this covalent linkage, these two strands are free to twist around each other. And if they're over-twisted, that will again go back in the opposite direction. If they're under-twisted, they'll go back and form the normal twist. So the normal, you have 10 base pairs, 10.5 base pairs per turn. So all this does is it gives you basically a free end of the DNA which can rotate around itself. And since twist and writhe are interchangeable with each other, this is basically just the exact opposite of the helicase. The helicase is pulling those two strands apart. Once they've been pulled apart, then you nick the DNA, and just nicking the DNA is breaking that phosphodiester backbone. It can then spin around by itself. Then the topoisomerase reverses this reaction. Now your tyrosine still has an OH, and you've reattached your DNA to itself. So topoisomerase I, very common. You see it all the time. There's also topoisomerase IIs. These are, in fact, much more complicated enzymes because what they need to do is break two strands and pass another strand to them. So it's got this writhe problem where you've got double strands which are crossing over each other. And to remove one of those double strands crossing over each other, you've got to break in both strands of the DNA and then pass one of the strands through them and then back on the other side. This process, unlike the case for topoisomerase I, completely requires ATP, whereas the topoisomerase I reaction does not. So this turns out you have mostly topoisomerase IIs in replication. There's another example of where you need topoisomerase IIs that we may get to at the end of lecture today. So topoisomerase II or I doesn't really matter which one you use. Either one works. And that's just for getting rid of all that extra supercoiling which happens because you pull the two strands apart with helicase activity. Yeah?
So does it cut both strands, or does it just cut one and then move the other one further?
So topoisomerase II cuts both. Sorry, the question is, does it cut one strand or two strands? So topoisomerase I has this covalent linkage which forms and then reverses. And that's just with one strand, because it's one tyrosine that does that. In terms of topoisomerase II, it cuts both strands, because you've got to pass both strands through each other. So it has to cut both strands and pass them through. In order to do this and make sure that you don't have all kinds of problems with your DNA, you also have to bind very tightly to those ends. And that's what's supposed to be shown here in the middle of the slide. Yeah, question in the back. Was there? OK. Oh, yeah, sorry. So the question is topoisomerase preferable to topoisomerase I versus topoisomerase II relative to the amount of ATP which is used? It doesn't seem to make a huge difference. There are, again, in this particular case, it doesn't matter if it's topo I or topo II, just in terms of getting rid of supercoils. But we'll see once we get, again, hopefully to the end today, when you replicate a circular genome, and most prokaryotes, bacteria, and archaea have circular genomes. If you replicate a circular genome, you end up with two genomes that are attached to each other, like this. Then you have to have a topoisomerase II to separate them relative to each other. Yeah, question right in the back. Okay, so the question again, I'm going to paraphrase, is how often do you have these topoisomerases and how often are they being used? Are you constantly having topoisomerase activity in terms of just controlling the amount of supercoiling you have inside the cell at any given time, or is it just in particular places and times? So the answer to that is it seems to be that the topoisomerase activity is constant, but it's wherever that topoisomerase happens to be. So if you think about replication forks, there's a topoisomerase at that replication fork. There are topoisomerases that are involved at the end of replication when you got these DNAs which are attached to each other. You also have topoisomerases that are associated with the domains, the loop domains of chromatin, which are supercoiled relative to each other. You have topoisomerases really at the base of those. So basically, where you need them, you've got them. Okay, so continue here. We mentioned the fidelity that you get from checking base pairs, which is what you get, just the geometry of the appropriate base as it comes in. That's the fidelity you get from the DNA polymerase. I'll talk about proofreading if it's the wrong nucleotide that actually gets incorporated, but that only gives you about one mistake in 10 to the 7, 10 to the 8th. People argue about the exact numbers here. And that gets me to, somebody asked me the question in office hours, Dr. Stedman, do you expect us to remember exactly 10 to the 5, 10 to the 3, 10 to the 2? In terms of the fidelity, no, because people argue about it. But knowing the relative amounts that you have, for instance, the polymerase itself is the one which has the most contribution to your fidelity versus these other mechanisms I would expect you to know. So here, polymerization, 10 to the minus 5. Your proofreading, 10 to the minus 2-ish. And again, people argue about that. How do you get that other 10 to the minus 2, 10 to the minus 3-ish? Mismatch repair. And so how does mismatch repair work? So this is if proofreading doesn't work, and you end up with base pairs which don't match each other. And the polymerase is cruising along pretty fast. You saw in the animation that's actually slowed down a huge amount relative to reality. It's probably better to slow it down even more if you want to be studying it. But there are these mismatches that happen, and that's basically shown up here. So you've got this mismatched base pair which has happened. How do you deal with this? So these are the so-called mismatch repair proteins, and they give you that last little bit which gets us to your normal replication. How do we know about them? Mostly from the awesome power of genetics. People made mutations in E. coli to start with, later on in other cells, and they found that these particular cells made way more mistakes, 100 to 1,000 times more mistakes, than cells that were not mutated. And then they figured out what that gene was, and they tried to figure out what that gene product actually did. And so that's how these were originally found. So anytime you see something that says MUT-S, MUT-L, anything like that, MUT-H, that's just the geneticist's way for saying you make a mutation in it. The phenotype is you have a mutator phenotype, i.e. you make way more mutations than you would otherwise. So these are the proteins which then first bind to one of these mismatches. How do they know it's a mismatch? Just the same way that proofreading works. The geometry is wrong. You don't have your two sugars in exactly the right place. The two backbones are in slightly different orientations than they would be as if you didn't have a normal AT or GC base pair. So they just recognize the DNA as being different. That's in fact shown down here in the corner. Here's one of these different DNAs, i.e. you have a mismatch of the bases in the middle. That gets detected by this MUT-S protein. Once you have this MUT-S protein that sees one of these problems, it then binds to another protein, which is this MUT-L protein. What that does is it makes a nick in the DNA, leading to removal of a whole strand of DNA that had that mismatch in it. That release now, you've got a 3'OH, you've got a single-stranded template, there's plenty of DNA polymerases in the cell, they will now bind to this and repair it. Of course, the key to this is that you find the right one of those nucleotides that were in the incorrect base pair to get rid of, because there was one that was correct and one that was incorrect. Which one's the incorrect one? It's the one that was newly added. How do you know which one is new? Well, there are a couple of different ways to know that. One of them, this is what's used in E. coli, but not in eukaryotic cells, that is that you have methylation that happens on your DNA, and that methylation happens on old DNA. So your old DNA is methylated. Once you've undergone replication, which is shown here with your replication fork, you have now one strand which is methylated and one strand is not methylated. It's also known as hemimethylated DNA, hemi just being half. So one strand of DNA is methylated, the other strand is not. That means it's newly replicated DNA. The methylated strand says, hey, this is the old strand. This is the one we've checked already. Whatever's on that new strand that doesn't match here, get rid of it. So that's the way that bacteria know which is the new strand and which is the old strand, so that mismatch repair only takes out the new strand and repairs the new strand. Well, what about eukaryotes? How does that work? Well, the way that eukaryotes do this is they look for nicks in that strand. Now, why do you have nicks? Nicks is getting back to the question we had a little bit earlier. How do you get that ligation to take place between the 5' phosphate and the 3'OH at the end of each of your Okazaki fragments? Well, okay, you end up having the ligase connected to that sliding clamp. But that nick says automatically that's the new strand. That's the new strand which you've just made. So that works great for the lagging strand. What about the leading strand? Because that doesn't have these individual Okazaki fragments. And that gets back to the question before about pCNA on the leading strand. It turns out that you do have a few nicks on the leading strand, where you do have to have reassociation of your polymerase and reassociation of that sliding clamp. So you end up with nicks on both strands of those newly replicated pieces. Yeah? So the question is, this seems to be a really inefficient process. Again, I'm just paraphrasing your question, that you should be repairing much better on lagging strand than you are on leading strand, and that's the prediction. People have in fact seen that. You do get more mismatch mutations that happen on leading strand versus lagging strand, but it does seem to be much, much less efficient. And the answer, the best answer that I know of, they actually discuss it a lot in that paper if you're interested. That's why I gave you the link. But the answer basically seems to be evolution's inefficient. It takes what works and not necessarily, and so it's going to be a balance between how many mutations you end up getting and how efficient your repair processes are. Now, again, that's a real hand-waving answer, but that, as far as I know, is the answer, the best answer at this point in time. Okay, so the last couple of minutes I want to talk about initiation. And if you have the notes, so wow, he's got like a ton of slides he needs to go through. A lot of them are basically exactly the same. So this is great and wonderful. We've talked about replication forks, but you've got to start replication forks somehow. And where do you start? That's just the origin of replication. So origins are where you have that whole replisome that gets assembled. We know it's hundreds of nucleotides in length, each of those, sometimes as much as thousands. Always AT-rich. Why do you think having AT-rich, i.e. lots of AT base pairs rather than GC base pairs, why would that be good? Easier to separate the two strands relative to each other. What interacts with them? In initiator proteins that we'll look at in just a second. Those initiator proteins basically lead to getting the two strands apart. And the key is getting those two strands apart. Again, why AT base pairs are really important. Once you've got the two strands apart, you're going to get your repliposome on there. Everything is going to go. So the key is getting the two strands apart in the first place. So what happens once you've got the two strands apart? Then you've got your primase. Those primases lead to extension. As soon as it's long enough, you'll get your lagging strand. Whenever you pull two strands apart, you're always going to have two replication forks. So there's a replication fork here and a replication fork here. Each of those is going to have a leading strand and a lagging strand. So whenever you open them up, you're going to have two of those. Let's look a little bit at how you go about doing this. Again, the key is getting those two strands apart. How do you get those two strands apart, and where do you pull those two strands apart? This has to do with the initiator protein, and I'm just calling it initiator protein now because this is general for all replication of double-stranded DNAs. We'll talk about the individual ones in bacteria and eukaryotes in just a second. So initiator binds. It binds, as far as we can tell, relatively specifically. So it's going to bind to specific sequence in DNA. We know this is true in bacteria. Seems to be true in yeast. Eukaryotes is a little less obvious. Once that happens, you now have DNA unwinding that takes place. What unwinds DNA? Helicase. And so you have to have this helicase brought into the appropriate place. And so that's recruitment. It's protein-protein interactions between this initiator protein and the helicase, which is there. It's in fact best known in E. coli. Turns out in E. coli you've got one and only one origin of replication in the whole genome. It's called the chromosomal origin of replication, or ORIC. Not surprisingly, that's where initiation factor is bound. You get the whole process together. End up with two replication forks that go all the way around the genome in 30 to 40 minutes. Depending on your strain of E. coli, that's 4.5 to 5 million base pairs. So in 30 minutes, you've unwrapped about 500 million twists. So clearly you need topoisomerase, and in fact some of the best antibiotics that are out there are topoisomerase inhibitors because they'll stop this replication actually from taking place. If we zoom into that ORIC in a little bit more detail, now we can see this origin of replication, again, AT-rich sequence and a binding site for your initiator proteins. What are those initiator proteins? They're DNA A. So initiator binding protein E. coli, DNA A. DNA B we've already talked about. That's the helicase. That's that very first step. What do you need to do? You need to get the helicase onto the DNA. Once you've got the helicase on the DNA, you're good to go. Everything else will happen perfectly normally. So getting the helicase on is absolutely critical. In eukaryotes, this process, getting everything together, just like DNA A, is something called ORC. And no, it has nothing to do with Tolkien, Hobbit, etc. This is the origin recognition complex. So complex should tell you a lot of different proteins together. What do these things look like? They bind to DNA. We now have structures for all of these. This is from a couple of years ago. DNA A binds to a whole stretch of DNA. Each of these individual gray and blue pieces here is one DNA A molecule. The ORC complex, these are 3 different proteins, also sort of a wrap around the DNA. Turns out archaea and eukaryotes actually have extremely similar proteins that are doing this job, and then there's some really funky viruses that do strange things as well here. One of the issues that you have with replication is you only want to do it once. Because if you're having replication forks starting all the time, you'd have a problem. Because once you've gotten to the end of replication, you need to separate your 2 chromosomes, right? Because they're going to go into new cells. Well, if you're replicating all the time and starting replicating all the time, you've got a big problem because you try and divide your 2 cells, but the DNA is still replicating. And so how do you get these things separated from each other? And so this leads to a concept called licensing. And licensing basically says, hey, I've replicated here. Don't replicate here again. And if you think about it, it's DNA binding proteins, right? That's what DNA A does. DNA A binds to a specific sequence, the ORIC, on DNA. Why shouldn't it keep binding there and keep bringing in the helicase? Well, the way that happens actually is exactly the way that E. coli knows which of those strands is the one to replicate in mismatch repair. Old DNA is methylated. New DNA is hemimethylated. Now, hemimethylated, one strand is methylated. Basically, this is the little flag that says, hi, I just replicated. Don't replicate me here anymore. And once it's been longer, then you get methylation. So once you've done your cell division, now you can replicate there again. And so that's the way that he works for E. coli. For eukaryotes is considerably more complicated. Let's take a quick look at it. For eukaryotic replication, eukaryotes in general have ridiculously large genomes. That's like our genome. 3 billion base pairs in just one copy. We've got 2 copies, 6 billion. How the heck are you going to replicate that whole thing? And it turns out that the polymerases in eukaryotes are actually a lot slower than the polymerases that you have in bacteria. And you've got these annoying nucleosomes that you've got to deal with. So it turns out that replication in eukaryotic cells is really, really slow relative to what's going on in bacteria. So how do you deal with this? Instead of starting in one place, you start in lots of different places. We know what those places are in yeast. These are what are called autonomous replication sequences. And we may get a chance to talk about those in just a second. But basically, if you take this particular sequence, put it into a piece of DNA that otherwise would not replicate, it now does replicate. And that replicates totally normally. And so that's the way these things were originally identified, is taking a particular piece of DNA, which is not surprisingly a binding site for work proteins, put that piece of DNA into other piece of DNA that replicates just normally. Take them out, they don't replicate anymore. So that's the autonomous replication sequences. In larger eukaryotes, all my yeast biology friends keep reminding me that they're not higher eukaryotes, they're just larger eukaryotes. You have these clusters of replication origins, anywhere from 10,000s to 100,000s of nucleotides. Just like you have in bacteria, this is a bidirectional replication that takes place from one particular position. And again, very often they're regions of the chromosome which will replicate at the same time. How are they replicating at the same time? Well, they all replicate in S phase. Hopefully, cell cycle is review for all of you. S phase is where you have replication taking place. Turns out that with all the number of replication origins you have, 1 hour would be plenty. You've got 8. Why do you need 8 hours to replicate in S phase? That's because of our friends U and heterochromatin. Euchromatin replicates really fast and really early. Heterochromatin takes a while because you've got to get all of the other proteins away from that. And we know that from looking at experiments like this. Here's one particular chromosome. You look at incorporation of one particular nucleotide here, which you can label. The euchromatin is replicated first, and then you have some of the heterochromatin and later parts. So different parts of the chromosome are going to replicate at different times. Yeah?
Just going back a little bit, so you're saying that the eukaryote has DNA sequences that initiate replication? Is that what you mean?
So the question is, are there specific sequences that are used for replication origins in eukaryotes? And the problem is, eukaryotes mean a whole bunch of different organisms. So we know that in yeast, there's some very specific sequences. In larger eukaryotes, like human cells, it's a lot less obvious where those things are. Yeah, in the back.
So this timing, is this for humans? The 8-hour
Yeah, for most of these human cells, but again, it varies a lot. But that's generally the case here for those human cells. So I think what we'll do is we'll stop here, and I'll talk more about the origins after the midterm. So basically, where we got to today is going to be what's on the midterm. What's after this is after the midterm.